Yeah,
Okay.
yes, it looks quite visible. Thank you.
Okay. Sorry, I'm just trying to press got it from the meeting being recorded. Sorry, I think I have to turn off the laser pointer to do that. There we are.
So it says recording in the upper left. So it seems
It's okay.
like,
I think, yeah.
Oh, we have the laser pointer.
yeah, we got the laser pointer, I think.
Okay, very good. So yeah, so thanks again. It's really fun to talk today. So I'm going to talk about some old work and I'm going to talk about some new work. So sorry for the old work, but I think it kind of feeds into the new, so we have to go that way. Maybe I just begin by just quickly saying the focus of my research group. So like I'm sure many people in the room today, we're trying to use concepts from statistical physics. So my background is in condensed matter theory, and we're trying to use concepts and ideas and approaches to study emergent behaviors in biological systems. And the focus of the lab is on multiple different things. We're interested in the morphogenesis of epithelial tissues. We're very much interested in the mechanisms that regulate stem and progenitor cell fate decision-making, again, with an emphasis on epithelial tissues. We're very much interested in how injury can drive a process of natural cellular reprogramming and the role that that may play in the presence of oncogenes and early tumorigenesis. And lately, we've got very interested in the problem of epigenetic inheritance and biological memory. So I think what's a bit unusual about us is that over the years, I've transitioned from being a condensed matter theorist now to running a small wet lab. And our wet lab focuses on, it's small, it focuses on mouse genetics and organoid cultures. And we have lots of collaboration. So that's us. And what I want to focus on today is an example from morphogenesis of epithelial tissues. Now, I'm not quite sure how the online platform will work, but I'm obviously very happy to have questions as we go along, and we'll see how that goes anyway.
Yeah, I can
So
relay the questions that people.
Yeah, so we have
a
an audience in a room here, and we will just speak up without inhibition, I suspect.
Yep.
And then we have an online crowd, and they put questions in the chat, or even interrupt. We will make sure you get them.
Okay, so just to say, I actually don't think I see the chat, so I'm relying on you guys if something comes up in chat to do the shouting. Okay, so the problem we're going to look at today is branching morphogenesis. And as of course you know, to sustain life, all organisms have to exchange nutrients, chemicals, waste products with the environment. In single cell organisms, the surface area of the cell is sufficient to do that. But obviously in multicellular organisms, we have to find strategies to maximize exchange surfaces. And this is achieved in different ways. So for example, in the small intestine, the epithelium has these long protrusions into the gut lumen known as villi, which increases the surface area for exchange. And of course, in three-dimensional contacts in other organs, these exchange surfaces are packed around complex, highly ramified branched epithelia. So you can think about kidney, lung, the mammary gland epithelium, pancreas, and so on. And so the question, the ambitious question that I want to begin to address in this talk is how can information that's encoded in the genes, gene products, and at the cellular scale translate into this large-scale organization and function at the organ scale? You know, how can we get insight into this problem? So the problem is exemplified by a first canonical system that we studied some years ago now, which is the development of the mouse mammary gland epithelium. So in the mouse, the mammary gland initiates as placode-like structures along the ventral epidermis. And those placode-like structures form a bud in the mammary fat pads. The mammary fat pad is a two-dimensional adipocyte-rich tissue into which this epithelium is going to grow. And by around birth, the epithelium has formed this small rudimentary tree-like structure. And then postnatally, this rudimentary tree grows through a series of tip bifurcations and elongations into a complex ramified epithelium. And this epithelium then undergoes bouts of growth and regression in adults, which gets fixed in pregnancy and generates these milk-producing units, which are the alveoli.
Ben, so the milk-producing units are at the tips. It's not produced elsewhere and then pumped along the.
Yeah. So the way to think about that, and you'll see it in a second, is you've got this very filamentary ramified epithelium, and then from the in the adult, you have bouts of growth and regression spontaneously from regions along the epithelium. So you get little outgrowths and then regression. But in pregnancy, those outgrowths become fixed as these little alveoli-like structures. Okay.
Okay, and then the alveoli are themselves composed of many cells, or are we just, this is not one gigantic cell, this is some.
Oh no, sorry, no. So this is macroscopic. You will see.
Okay,
to give you some sense of scale, well, here's a sense of scale. Okay, so if we focus on the developing the pubertal development, the branching morphogenesis, here's a typical tip. So all proliferation is contained in the tip, and you may have 200 cells or so in the tip, and those cells fall out of the tip and give rise to a simple stratified epithelium comprised of myepithelial basal cells and luminal cells. Okay, so these cells in the tip are termed mammary stem cells, and these cells are driving a process of growth, bifurcation, and elongation.
I see.
Okay.
I see. And there's a different gene expression pattern for each one of these cells, and that's somehow maintained during this growth process.
It's a great question. So we're not going to dig too much into that, but in a way, let's keep that thought. If you look at the ductal cell types, then you can identify clearly 3 different cell types: the myepithelial basal cells and ER positive and negative luminal cells. But within those compartments, there are even more subcompartments. And the question of which cells here generate those cell types is itself a question.
Okay. And then we can think of these collections of cells as viscoelastic fluids. Maybe this is coming, and I should just wait.
Well, that's another very nice question, and it may not come, but maybe I don't want to deal with that just now.
Okay, fine.
It is a nice question. Okay. All right. So here is here's an example. Here are some examples of the fat pads. So this is at 3.5 weeks. What you can see in yellow is the outline of a fat pad. And what you can see stained in keratin 14, a basal cell marker, is that growing tree. At 5 weeks, the tree has grown a little bit more. And by 8 weeks, it's filled the fat pad with this ramified network. Okay, so those are snapshots of the growth process. One other thing to note is that if we stain the tissue if we incorporate EDU, which is a thymidine analog, it gets incorporated during DNA synthesis, then you can see that all the proliferation is really confined to the growing tips, right? So when the cells leave the tips, they fall out of cycle. Okay. So the question I want to address is, how do these mammary stem cells, how do they integrate fate choice with all of these collective cell movements to generate this ramified complex network? What is the identity of these mammary stem cells? What's their multiplicity? And what is their lineage potential? Are they all able to generate basal and luminal cells, or are they restricted? And are these programs that we can abstract here, are they conserved in the development of other branched organisms? Is the strategy the same?
Can I ask a question? So the adipocytes, the fat cells, where do they, are they also proliferating, and do they also migrate or move around when the branching happens?
Yeah, great question, and that will be actually in a way fundamental when we get to this this last part, if I get there. So in the case of the mammary gland development, there's not much proliferation in the fat pad. It doesn't have to expand very much. The ductal morphogenesis takes place into this preformed fat pad. There are cell movements within the fat pad, but it's largely preformed. But that won't be true in other developmental contexts. It's an unusual, in that respect, it's an unusual tissue. That's a good question. All right, so how should we begin? If we were molecular cell biologists, we would want to identify the repertoire of genes, gene products, signaling pathways, and start from a very microscopic type of description. But I'm a physicist, and most of the people on the call are from physics. So we would naturally think that maybe we should start with a large-scale organization and see whether there's some principles that might emerge from that. So that's what we did. Okay, so this work, this is several years ago now. This work was carried out in collaboration with Jacob van Rienen, who at that time was at the Hubrecht. He's now in Amsterdam. And the grad student doing that was Kalinda Sheila. She did all of the heavy lifting. So here is a mammary gland fat pad from the mouse. The blue is a DAPI stain, a nuclear stain, and the white here is keratin 14. So what Kalinda did was that she segmented this mammary fat pad, and she did that for many fat pads. And then she constructed the network, the topology of this network over many different glands. And what you can see is that some of the trees terminate rather early, and other trees go through large numbers of rounds of branching. So there is enormous amount of heterogeneity present in this network. Some of the branches go through 20 to 30 rounds of consecutive branching, while others may terminate after just a few. Okay. So the second thing to note is that the frequency of TEBs, these are terminal end buds. TEBs are tips. In the field, they're called TEBs. So the frequency of proliferative terminal end buds diminishes throughout puberty. So early on, there's lots of active tips. By the time you get to mid-puberty, there are lots of tips where all of the cells have stopped cycling. And by 8 weeks, there's very few. So where does this complexity come from? Does it come from having mammary stem cells with a variable amount of proliferative potential hardwired from the beginning? Or what seems much more likely are extrinsic influences controlling the effective fate behavior of equipotent cells, right? So is it environmental factors that's really creating this heterogeneity?
So maybe this is one of your two possibilities, but could it be that these proliferating branches are secreting a chemical that downregulates the proliferation of things in neighboring tissue somehow?
Yeah, you're absolutely on the right track, and I think, David, I think you'll guess the answer, before I get to the answer. So let me press on, but you're absolutely on the right track.
Thank you.
Though I would place that in environmental factors, but okay.
I see. Okay, good.
So the first thing to note is if you look at the widths of the ducts with branch level, or the length of ducts, the length between consecutive branches, these are average values, then they're remarkably constant over the branching level. I mean, I hope the level is clear. You know, the first branch is branch level 1 and then 2 and so on. And the second thing is that even though tips are falling out of cycle, if we look at the proliferative activity of the tips that remain in cycle, they remain roughly the same over time. So all of this suggests that the process is equipotent. It's suggesting that the tips are just as potent at 3 weeks as they are at 5 weeks, as they are at 8 weeks, if they don't fall out of cycle. Okay. So what's the source of the heterogeneity? So at that time, I had a fabulous postdoc, Edouard Hanezo. Edouard now has his own group in Vienna, at the IST. And so Edouard suggested, let's do the following thing. If it's equipotent, every event can be classified according to a bifurcation or termination. And so let's suppose that there's a probabilistic rule for that, which will assign a probability Q, which will change with branch level. And let's see what happens to that probability over time. So this is absolutely empirical, absolutely empirical. And if you measure that, then early on, this probability is very small. It means that most of the branches branching is symmetrical, but this converges to around 1/2. So 1/2 is like saying, let's flip a coin. If it's heads, let's bifurcate. And if it's tails, we terminate. Okay. Well, obviously you can do this, but it doesn't mean that the statistics of this network matches that, right? So that doesn't have to follow, but we can test that. We can say, let's use this as an empirical rule and generate networks and see whether they are similar to what we see. So first of all, if we look at the fraction of tips that remain proliferative over time, then the black line, which is called here model, it's an empirical model. It's just taking this Q value and running it out. It predicts pretty well how that fraction diminishes over time. And if we look at the statistics of the subtree persistence, the chance that a tree persists to a generation, or we look at the distribution of subtree sizes, it works pretty well. Right, so that's just taking this empirical rule. It actually predicts pretty well the statistical distributions that we see.
So Ben, do you have an understanding of why Q is actually increasing with time and that sort of thing?
Yeah,
I mean, it's a collective of cells. So
I do. I got a challenge for you, David. Given your previous question, you're going to be able to guess what it is in a second, right? So here's the question.
I'll be quiet for a while.
No, I'm not kidding. I'm not kidding, right? So what we find is the branching topology is predicted by a simple statistical rule that's acting at the ductal scale, right? So if we just think of these tips as single units, it looks like there's a simple statistical rule that's governing their behavior. And in fact, the origin of this that we figured out just
So then there's someone else is going to harass you.
No, sorry, I just have a quick clarification. So between each probabilistic branching or not branching event, it's assumed that the branch extends by a fixed length. Is that correct? At least in the simple model that you showed?
No. So here it's purely topological.
Oh, okay.
Now it's going to turn out that the timing between consecutive branching events is actually random. Okay. It's statistically uncorrelated. You'll have a nice Poisson distribution, but forget about that. This is just about the topology.
I see.
Right?
Okay.
So this is subtree size in terms of branches. You know, this is a trivial model, right? It's like a birth-death process up here.
Sure. Right. Thanks. Yeah.
All right. So where does this come from? Well, I'll tell you the answer, and it's actually, David, it's very close to what you asked moments ago, right? So here's the answer. Okay. What regulates this near balance between bifurcation and termination? So if you look at the branching network, you can see there's very few crossovers. And if we look at the coarse grain density of the epithelium, it doesn't change too much. So there's very few crossovers, roughly constant density. So this is very much suggestive of a very simple kind of process. And the process is that tips can stochastically branch. They elongate at some rate. But when an active tip encounters a maturing duct, or more precisely, when it encounters factors secreted by a maturing duct, the cells fall out of cycle. Okay, so at this stage, this is a hypothesis. This is a hypothesis. So what does that look like? Well, you know, here's a little toy movie in a square fat pad, and of course, what happens is that you have this front of active tips that leave behind a network of inactive ducts, right? So all of the activity gets localized towards the periphery. Okay, so this is a hypothesis, the cartoon, right? So can this model predict the network statistics? Okay, so here's simulation. Here's another example of the experiment. Interestingly, and it was very surprising for us, that in our little toy model, the distance between consecutive branching would be exponentially distributed because it's just a stochastic process, right? But actually, experimentally, if you look at the branch length distribution at two different time points during development, it is actually remarkably exponential. So it really looks like this branching is a stochastic process. We don't understand why, right? So why should the timing between consecutive branching events be statistically uncorrelated? We don't know. but that's an empirical observation, and it actually fits very well with the toy model.
So this is actually a branching process in 3 dimensions, I presume. Your model.
Oh yeah, no, sorry, this is a great question. So it's a great question. So and we'll come to that. In the mammary fat pad, it's very quasi 2-dimensional. It's like a pancake. There's a little bit of 3-dimensionality. If you go back to this image, you can find crossovers, but it's difficult, right?
Okay.
It's really kind of 2-dimensional.
Okay. Thank you.
Yeah.
Another quick question. So in the real biological system, is the area of the fat pad kind of predetermined before the branching networks are generated, or it's
Yeah, it's a great question. So the fat pad is basically framed at birth. And although it will expand a little bit, of course, the animal grows and the fat pad will grow a little bit. There's not much growth. So that you should think of the branching morphogenesis taking place within a preformed fat pad.
Cool, I see. And another kind of follow-up question is, could you specify a little bit more on what's the biological functioning of those tubes? Like, is that to make those fats stored in the fat pads accessible to the animal, or what's the?
Oh no. So the origin of this branching is a teat of the animal. So the alveoli give rise to milk, and that milk passes through these ducts and comes out here. So the primary function of those ducts is to convey the milk, and that's why you have these myepithelial basal cells to do the pumping of the milk.
So we're seeing the teat, for example, in the upper left picture is
this is a
okay.
So this is a lymph node. The teat is around here.
Okay. I see. But they grow. They nucleate roughly where the teat will be, and then they grow out from there.
Yes. No. That's where they nucleate.
Thank you.
Yeah. This, by the way, just as a digression, this process is amazingly robust. You can transplant stem cells into an empty fat pad, and it will undergo branching morphogenesis.
Wow.
It's a very self-organizing system. So we take these simulations, we take the experiment, we've got these are typical. We've got many experiments, many simulations. These are typical networks, and there it is, right? This toy model predicts very well this average tip termination probability, the subtree size probability, the persistence. And then you can ask, well, but how many parameters have you got in this model? And the answer is that we've got no adjustable parameters. So the branching process, as you'll see in a second, just depends on the ratio of the branch rate to the elongation rate. And we measure that. It's a measured thing. So this is a 0 parameter
I mean, I agree with you, but presumably you fit an entire function Q of T to get some of these pictures, and it drifts up to a half, I guess.
No. So this is what the model. This is what the model will do. If you take literally that model and you let it run. So why should it start with a low value of Q? It's just space volume, right? When you start as a point, you've got lots of space, but then you end up on a 1-dimensional front. And okay. So you'll see in a second why we wind up at 1/2.
Okay, I see. I see you're generating the Q of T function as well as everything else.
Yes, absolutely. It's a self-organizing process. You'll see why in a second.
And if you were to do it in some hypothetical animal that had a 3-dimensional fat pad, probably doesn't exist. But if it did exist, would you get radically different results?
Just wait. Just wait. It's coming. It's coming. What about the growth dynamic? Okay. So we can go back to the literature, branching and annihilating random walks. There was a nice study by John Cardy and Uwe Tauber, but that's about a tip-tip annihilation. That's not quite what we want. But there was an earlier study by Cardy and Grasberger on epidemic models and percolation. And it's in this class that this system belongs. So why? Why is it an epidemic model? And it's easy to see. So this goes back to their, Cardy's early work and Cardy and Grasberger. So we have a reaction diffusion system where we can think of those active tips like infectious agents, which diffuse and they multiply. So the multiplying is branching. And as they move, they leave behind a trail of infected sites, but those infected sites recover, okay? So we've got inactive ducts. Those are the sites that have been infected, and they're immobile, right? So that's the elongation process. And these infectious agents are terminated when they revisit a recovered site. That's the tip termination process. Okay, so it's a kind of epidemic model. So they considered that model a long time ago. And what they wrote down is, so this is a Doi-Politi field theory. We're not going to go near that. But they looked at this field theory. So this field theory, the critical dimension is 6. So fluctuations important below 6 dimensions. And this model is kind of fun. It has a transition between a phase in which you annihilate with probability 1 to a phase in which the probability of escaping to infinity becomes non-zero, right? So there's a phase transition in this system. Now, in the biological system, we're very far from that critical point. So we're going to look at mean field theory. Okay. So the mean field theory of this problem is here. We've got diffusion, branching, annihilation. Elongation is generating the inactive tips, and then we've got annihilation. So if we make this thing dimensionless, so we've just got one parameter, like I said earlier, which is the ratio of the branch to elongation rate. And this dynamics is Fisher KPP. So this is a Fisher KPP reaction diffusion system. And what it's describing is a traveling wave. So let's look at this. Let's look at what happens over here. All of the active tips become confined to a front, and that front propagates forward, and it propagates forward as a Fisher KPP wave. So if we can look at that, we can look at that here. So if we just solve this equation, here you can see a soliton front of active tips, leaving behind a trail of constant density in active ducts. And that front propagates, of course, in dimensionless units. It propagates as the square root of our one parameter, the ratio of our branch to elongation rate.
So sorry, Ben, this is very beautiful, but there's 2 equations in the upper right. So you can see immediately that it's Fisher KPP by just taking the second equation and assuming it's in a quasi-steady state and substituting the first, for example.
Exactly.
Okay.
Exactly.
Lovely.
So now we can see the answer to the question, why do you wind up at Q equals 1/2? So if you think about what's happening at the front here, every time I bifurcate, on average, one tip pioneers new territory and the other collides with the trailing network. So by the way, David, this of course is very similar to your bacterial front propagation. Of course, it's, it's in exactly the same chapter of the book, by the way, right? It's a very similar kind of
We've heard reading the same book. Yes, right. Good.
Okay. All right. So this is mean field theory. What about experiment? So here again is a fat pad. This is data. Here's EDU. That's the proliferation marker. It's enriched at the front. And what Edouard did was to take many of these pictures and digitize them. And in fact, actually, this mean field theory works very well. It even predicts the ratio of the exponents of the soliton on either side of this active front. So it kind of works unreasonably well. Okay. Now we can't measure dynamics, but someone else did in a way. They just looked at some static pictures. This is not our data. And you can see that the position of that front, it's roughly linear, right? It's roughly changing linearly. In other words, it's consistent with a sort of constant speed of front propagation.
So just a technical question about the mice. When they're lactating, the milk is being produced at these tips, and then it goes to the tip. There's a pressure that drives it there. How does it get there?
Yeah, that's a really good question. So first of all, though, this is a really interesting question that a lot of people have studied, and we've not studied it. But so what I can say is that you have myepithelial basal cells that are able to provide that constriction, and that constriction, I think, is forcing the milk along the ducts. And how you do that in a coordinated way is a very interesting question. The other very interesting question, of course, is that if you generate networks like this, they're not optimized for distributing the pressure in the network, as you could imagine. But what I'm showing here is what happens during puberty.
Yes.
When there's pregnancy, there's a lot of remodeling. And that remodeling may be driven by fluid flow. I don't know. It's a really interesting question. Really interesting question.
Thank you.
Okay. So a second feature we can look at in this model is that, of course, you have chance events where you generate voids. So what we would expect to see is density fluctuations, giant density fluctuations. And interestingly, if you plot variance of particle number against average particle number, both this toy model and also experiment has this unusual exponent, an exponent which is not random, it's not 1/2. We don't have a mathematical argument as to why it's 0.65 or indeed whether it's 2/3. We don't know. But empirically, you get this anomalous type of exponent associated with these giant density fluctuations.
Can you calculate it from this theory that John Carty did, for example, or you could do it in 6 minus epsilon dimensions, maybe?
Yeah, I have a postdoc who's trying to do that at the moment, but not easily. And I should just comment, there's much nicer data than our data from Celeste Nelson's lab. So she followed up on this and actually, looked in with much more care. So the second thing, which had always been a puzzle in the mammary field, is how do you get directed growth? But of course, what we have in this system is that the dynamics itself creates a direction, right? You don't need any kind of morphogen gradient to instruct which direction you move. And so we can measure that angle of the duct orientation experimentally and in the simulation. And again, it works very well. So here we have directed growth without morphogens. It's just the pattern of the growth dynamic that produces the directionality. Okay. All right. So what is the identity of mammary stem cells, multiplicity and sublineage potential? And I'm not going to tell you in detail. I'm just going to give you the answer because it's a whole other talk and I want to get to new things. So what we can do is to do clonal tracing on the background, which is what Kalinda Sheila did. So here you activate a fluorescent reporter and then you see where the footprints of the cells are. So the principle here is that you label a cell and as it falls out of cycle, it leads behind these footprints and those footprints tell us about the decisions that were made at the tip. And what we find is that the end buds constitute a niche-like environment. And they support, so all of these cells have renewal potential. There's a lot of motion of those cells over time. And there are 2 types of stem cells. Actually, there are 3 types of stem cells in the tip, which are absolutely lineage restricted to generating basal cells or one of the types of luminal cell, right? So we've got 3 populations functioning independently as renewing populations that when the tip bifurcates, they duplicate and segregate. Right. So that's what's going on at the cellular level. Of course, no mechanistic insight here. But of course, we do have a platform to begin to develop mechanistic insight. So these cells within these 3 lineages are equipotent.
Can you say again, sorry, Ben, in the previous slide, can you say again what the thin blue cells in the upper right are? Those are.
Yeah, so the niche for these tips is comprised of many different factors. So these will include fibroblasts. There are also macrophages that accumulate around these sites that also produce factors. So we can think of this tip as like a stem cell niche. You know, one kind of analogy is in an intestinal crypt.
Yes.
You've got cells in the niche which stay put, and the cells migrate upwards out of the niche. Yes. Here it's the opposite. The progenies stay fixed, but the niche and the cells move into the tissue. but it's a similar kind of organization. I mean, you know, for physicists, it's very similar organization. Of course, the niche factors for the biologists, of course, those will be different according to the system. Right, so are these programs, meaning the large-scale programs, conserved in other branched contexts? So kidney development is much more complicated, but the early phase of development again occurs through dichotomous branching. And again, after an initial phase, which is very stereotyped, where you've got symmetrical branching, you then have a lot of irregularities. So here's a E16.5, embryonic day 16.5 embryonic kidney. And what I've marked here are 4 different subtrees, which are also marked here. And again, you can see that some of these subtrees are tiny, and some of them are huge, and everything in between. Okay. So is there a statistical basis to branching in kidney?
So can you tell us again what the function of this branch network is in kidney? It's not lactation. Is to get waste out of the bloodstream, or what is it?
Exactly. So the in this case, there's another phase of which is essential here, which is nephrogenesis, where these tips, when they finally complete this branching program, have to connect to the nephrons, and then again, this will be a system which gets rid of waste products and so on from the kidney. Let me, in the interest of time, okay. I'll be out of time. So it's a great question. It's not going to be a focus of
but by all means, press on. But it's only a soft boundary at 2 p.m. for us.
Well, I don't want to trespass on people's time. But let's continue. So in this case, again, we can look for evidence of equipotency. It's not quite so neat, right? So the average branch length is actually changing a little bit with generation number, although it flattens out. We still have this exponential-like distributions of branch lengths. We also have an asymmetry. There's differences in the aspect ratio here. Okay, but these are 3-dimensional. So we can play the game that David asked, what happens in 3 dimensions? So here is the simulation of branching in 3 dimensions. And here is a data set that was generated by Rosemary Samponia, who is a collaborator of ours. So she's in Colombia. And she had painstakingly reconstructed these embryonic kidneys. And here you can see what comes out of this 3-dimensional simulation. And this is what comes out of the experiment at E17. So we had several data sets across several time points. And now we can apply the same principle of branching, annihilating random walks. But now, of course, we've got a new parameter, which is how close do I have to get to stop? So in 2 dimensions, this is not a very interesting parameter because eventually they'll collide. But in 3 dimensions, it's a parameter of the problem. Okay, so we got one adjustable parameter and the rest we can measure. So here's that average tip termination probability for kidney. The purple shows the mammary gland for reference. So here you've got that stereotype phase of symmetrical branching. And eventually it turns into this more stochastic phase, again, approaching 1/2, but it rises because it's beginning to terminate. Okay. And if we look at now the statistics, well, first of all, let's look at that parameter. So if you think about the adjustment of this annihilation radius, this parameter, if the annihilation radius is very big, then all trees will annihilate before you get started. You launch this thing and everything annihilates on itself. It's all over. Then there's an intermediate regime where the chance of escaping to infinity is non-zero. And then eventually there's a regime where you inevitably have survival. The thing will grow out to infinity. And empirically, our parameter winds up around here. So we're in this phase where the system doesn't annihilate on itself. So once we fit this parameter, again, we can see whether we can predict the number of branches as a function of generation number, the subtree persistence, and again, the subtree size probability. across these different developmental times. And it works pretty well, right? It's, you know, there's nothing really to adjust here, just this one parameter, and it predicts pretty well all of these different distributions.
And the E, just a point of information, E17 is 17 branches or 17 days?
Oh yes, sorry, that's the biologist. So so E stands for embryonic day 17.
Aha,
It
okay.
means 17 days after fertilization.
Oh,
And
okay.
if we go back here, so at embryonic day 13, here's my little embryonic kidney. And then by E18, it's branched into this elaborated structure.
Thank you.
It goes on later, but we have data only up to E18. And after E18, you start to have different changes taking place in the tissue. Okay.
And some question. So here for the case of kidney, instead of have a confined space of certain shape, we are allowing it to grow freely in 3D, or it's the
Yeah. So I think I think I've heard the question, but if I haven't, please repeat it. I think you're asking whether the branching takes place into a kind of preformed background, or whether the background is also, you know, the mesenchyme is growing at the same time.
As I understand the question, it was more like, is it indeed 3D rather than 2D in this case?
Oh yeah, no. In this case, it's really three-dimensional. In fact, I should have had I've got a little spinny movie that other people have made. It's the way to think of it is like, and it won't surprise you, it's like a kidney bean. So if you can picture in your mind's eye a kidney bean, that's kind of the shape, right? It's kind of right, anisotropic, but very three-dimensional. Yeah, thanks.
But also, is there an analog of the fat pad? So does the mesenchyme provide the analog of confining?
Right. So here, you know, I get into terrible trouble with the biologists, and maybe rightly so. So in the case of the kidney, the mesenchyme is growing in concert with the epithelium. The two things are growing together, and actually there are distinct changes taking place in signaling factors and composition in the mesenchyme, and those must play an important role. So the way to think about this, and I think this is a very, you know, important remark, is, you know, what we're trying to do here is we're trying to ask, you know, if we strip away everything, what's the simplest possible description that captures a lot of what we see? There's a lot of details that are invisible to us here that are beyond the resolution of this data set, but which are terrifically important for the biology. And if I get to the third chapter, which is just, you know, like next slide, then we'll see where that really plays a role in another context. Okay. All right. So are these programs conserved in other branched organs? Well, we looked in the lab, we looked at pancreas. They're conserved in pancreas, or seems to be. But let's look at a sort of exception, because this exception will bring us to a revised model. So let's look at this as the third and final example. So let's look at mouse salivary gland. So in the case of the mouse salivary gland, so in this case, you have an early bud formation, and you have multiple clefts formed in a single bud. So you have a sort of diversification of end buds early on. And then you have a phase that looks more like branching morphogenesis, tips having a single cleft, branching, elongating, cleft, branching, elongating, and so on. And eventually, at the end of this process, the termination is marked by the formation of acini, so these secretory acini clusters, which finally terminate the branching process. Okay, so what you can see on the right is not our data. This is from Abigail Tucker's lab, and this is an explant. You can see that little, I hope you can see that little early bud at E12.5, and I'm going to try and run the movie, and you'll see how it kind of elaborates. Is it going to go? Yeah, there it goes. I hope it's working for you too.
Yes.
It might be slightly hard to see.
Wow.
But you can see that inflation. And I hope what you can see is that the mesenchyme is growing and remodeling at the same time. So you have this beautiful kind of flowering of this branched tissue. All right. So we set about looking at this system in the lab. And the reason we did this is because this person, Lamonia Chutselli, who's a postdoc in the lab, she brought this project to the lab. And what you can see here is one of the salivary glands in the mouse, the submandibular gland, at E18.5 when the acini have formed. So you can see these kinds of circles here, perhaps. Those circles are multicellular units, which are the acini. And what she's marked in pen, or on the confocal image, is that she's traced out these ducts. And so what Lamonia did was to reconstruct the branch network of this submandibular gland. Actually, she's looking at one lobe of this submandibular gland. So at embryonic day 14.5, you have this tree-like structure. And you can see it here in XY, and here you can see in XZ. It's kind of, again, pancake, but it's more three-dimensional in this case. It's like it's clearer here. So at 18.5, you have this very ramified branching structure, and it's, again, quasi two-dimensional, but now you really do explore that third dimension. Okay. So she reconstructed these from multiple glands, which was a very, we tried to automate it, but that didn't work very well. You know, you're trying to use clever imaging tricks and so on, but in the end, manual counting was the only way. And again, here are the trees. This is 14.5. Here it is at 18.5. And it all looks like the usual story: small subtrees, large subtrees, everything in between. Okay. But now things are slightly different. So if we look at the duct length distribution, the distribution of duct lengths, before in the previous examples, it was exponential. Well, you know, it's kind of exponential, but there is this kind of fat tail here. And the other thing you can see is that at the early time points, the ducts are actually larger than they are at the later time points. That's, you know, that's important to know. If we look at the average duct length, it's also not flat. It also is traveling. Look, this is a log scale. So these are real changes. The duct length is changing with branch level. The width isn't changing so much. So the first thing we did is to say, well, you know, let's just measure this terminated fraction over with branch level like we did before, and ask, can we just take this model and predict the subtree size distribution or subtree persistence? And the answer is no, it's really bad. Doesn't get even close. Okay, so this very simple paradigm of birth, you know, death, termination, it's not working here. Okay, so now the second thing that we can see is that if we look at the gland itself, the gland is actually expanding exponentially rapidly. Well, this is the area, this is the whole gland here, it's very visible, but even that first lobule, which is what she segmented, that's also growing exponentially. The lines here are exponentials. Now, you should contrast that. If it was this branching and annihilating random walk, the front travels at a constant speed. It's not exponential like. Yeah. So there's a kind of inflationary process taking place here. Okay, so we shouldn't have expected this model to work well, because that branching and annihilating random walk model would give us a linear growth characteristic. So what's going on? Well, what do we think is going on? Well, what we can see, again, if we stain the tissue with a proliferation marker, we can see that submerged tips here are slightly less proliferative. That's quantified here, but they're still proliferative. So once you get submerged, you don't fall out of cycle, still proliferating. Well, okay, you're still proliferating. So what we think is going on, so the theory here was done by Ignacio Bordeaux. He's starting his own group in in Chile in the summer, still with me now. So this is work that Ignacio did. So he made the following hypothesis. We know in this case that the mesenchyme is growing. So let's suppose that the mesenchyme grows in a kind of affine way. And what it does is that the epithelium is expanding in response to that. So we've got the usual process of duct elongation. We've got tip bifurcation. But if we have inhibition due to crowding, then as the tissue expands, that tip can reactivate. Okay. So in this particular case, it's going to turn out that those tips never really stop. They just get delayed. Right. So you get a crowding phenomenon, but then as the mesenchyme around you expands, more space appears, and now you continue to grow. So does this inflationary branching arresting random walk? So it's obviously in the same chapter as the previous model, right? So no inflation is the old model, with inflation is this. Does this predict what's going on? So what are our parameters in this case? Well, first of all, we've got the expansion rate, which is exponential. We can measure that. Then there's the branch to elongation rate as before. We can measure that. We've got the branch angle distributions in this case. We can measure that too. But what we can't measure is the inhibition radius, the radius at which you get delayed and restart. And that for us is a fit parameter. So if we take this model, then this is, so in this case, we seed from 14.5 because prior to that, it's stereotypic. We seed from 14.5, and this line comes from the best fit to that one parameter, and that's the branch probability. So you can see this kind of inflationary branching process. But what about the other statistics? And if we look at the other statistics, now we can explain the subtree persistence, the subtree size distribution, the distribution of duct lengths. It's a little higher. Remember, this is a log scale, but we have no adjustable parameters here. And we also get, again, giant density fluctuations, which seem to follow towards a slightly different exponent, if exponent it is at all, which of course it might not be.
So Ben, when you say seed from 14.5, that's an initial condition for this more sophisticated model.
Yeah, so this is a really important point, because this is a really important point. So that very early phase of branching down here on the left is a diversification process. It's not a branching morphogenesis. If you look at that early duct, it's got multiple clefts that all form at the same time. And then it sort of branches out into multiple different seeds. But by 14.5, we think the system has then settled. And from then on, we think we're in this state. So we're using that as our initial condition, and then looking to see whether we can predict the 18.5 gland. So just to give you an idea of scale, I think the best one to give you an idea of scale is here, right? This is what the gland typically looks like. It's statistical, but this is what it would look like at 14.5, and this is what it looks like at 18.5.
And the model produces this aspect ratio, the fact that it's squashed a certain amount, does that come out of the model?
No, so that is hidden under here, the branch angle distribution. Once we measure those distributions, if we put that into the model, that comes out, right? But we don't, how can I say, we don't cook it up. We just measure those distributions, and that's what seeds the model.
So how do the salivary glands know to stop proliferating exponentially? Or what turns this off?
Oh yeah, it's a wonderful question. So why doesn't this just go on and on and on, right? I'm going to give a first evasive answer, is that the differentiation into acini is a terminating event. So these tips turn into differentiated acinar cells, and that's a terminating event. Okay, so you can ask, well, what tells the tissue to now form acini, right? And I don't know the answer to that question. I suspect that that there are different things that can contribute, and maybe they all contribute. So one thing that can contribute are hormones, right? So this is around the time of birth. So this is a point where you're going to actually start having to use this organ in a functional way. So there may be hormonal changes that instruct those cells now to differentiate. But the second thing, which is very interesting, is that in addition, there are mechanical changes that are occurring in the mesenchymal populations, the kinds of things that Celeste Nelson and others are very interested in understanding, and they may play a role in driving those cells towards differentiation. So there may be chemical changes, mechanical changes, hormonal changes that instruct that termination event.
So again, the acini at birth start producing saliva, which then gets coming out, squeezed out of this duct by some contractile process.
Exactly so. Yeah, exactly so.
Great.
Yeah. All right. So so there we are. And then finally, because it's a physicsy kind of audience, so now we can go back to our mean field theory, and we can put in some new terms. So now we need to add stalled tips alongside inactive ducts and active tips. We need to put in the expansion rate in dimensionless units here, that's gamma. And then we have now an advective term due to the expansion of the tissue. So this mean field dynamics, if we have no expansion, will collapse back to the original Fisher-Capey p-type model that we had before. And in this case, if we ask what happens in this case, so here you can see the sort of numerical integration of this equation. Obviously this thing grows exponentially rapidly. So I've taken the radial distance in dimensionless units and contracted it by that exponential, right? So we're in the kind of co-traveling frame of the net expansion of the tissue. And what you can see is that in the middle of the tissue, we have a constant density of active tips, of stalled tips and ducts. It's all, each of these is normalized to unity. That's because the tips are constantly activating even in the center of the gland. But what we see is that there's a peak in the active tips at the front as it propagates. And in fact, if you look experimentally, although this might be a bit of an over interpretation of the data, we can actually see, if we look at the proliferative index, that there is actually a peak as we move towards the edge of the tissue. Whereas if we look at the proliferative index of the mesenchyme, it's roughly flat, which is consistent with that exponential-like expansion of the tissue. Okay. Fine. The reason that Lamoni is doing this, and I won't go into this, so Lamoni is very interested in understanding the mechanisms of cell fate specification. And so what you can see here are clones induced on that tree. So she induces clones at 13.5. This is an 18.5 tree, and each of these different colors is a different clone. And she's been able to show that you start with multipotent cells that then segregate inside out into bipotent cells, which are restricted to myepithelium basal lineages or into luminal acinar lineages. And eventually those split up into 4 lots of unipotent cells. So I won't go into this, but this is her focus. And that kind of lineage segregation takes place during the branching morphogenesis. We can also see that in the single cell data, where you see, this is single cell RNA-seq data, where you see those multipotent cells splitting into bipotent and then splitting into these unipotent populations. All right, so I'm going to ask Farzan how much time, because I'm done, except that I wanted a few kind of summary words to close. And I don't know whether, I don't want to trespass on people's time.
I would love to hear the summary words, please.
Okay,
Yes.
Okay. Well, blame David and Farzan. Okay. So are there alternative space filling strategies? Of course. So first of all, the first point to note is in this branching annihilating random walk type paradigm, you've got very inefficient packing, right? So you've got this stochastic self-organization, but you have these very large voids. But the big advantage is that you can generate these networks using unchanging and local rules, right? It's a local measure to decide what the next move will be. Now, of course, you can do way better by having an evolving branching rule, right? You can get a much higher density, but to achieve a much higher density, you've got to evolve the rule over time. And that brings an overhead in a biological context. So are there contexts where you're willing to pay that overhead? So that brings me to, this is mouse lung branching, and I won't go into details. Of course, I won't go into details, but here you have very stereotyped programs of branching and side branching with Turing-like instabilities generating those, and you have these beautiful orthogonal-like structures that bring these highly stereotyped patterns. So there are places where you really need to get this right. But interestingly, we've been segmenting, or we as a shared student of mine with Alain Chateaudal in Paris have been segmenting human fetal lung. And there it looks like you have a much more stochastic type of dynamics during this phase of tip-driven branching morphogenesis. So we're digging into that. It's really exciting. So this here, it seems that you really do have a similar kind of branching paradigm to what we've seen before. So it seems that in human, you don't have to be quite so stereotyped as you are in mouse. Other people have proposed different kinds of models. People in the kidney, people have proposed mathematical types of models, you know, where you've got conserved types of mathematical rules, or this half delay model, where you have a sort of pause of one of the tips before it can branch again. So in our hands, these models struggle to reconcile the high degree of variability that we see in the embryonic kidney, but they, you know, but they could be correctly describing what's going on early in the branching program. And then a kind of final remark, and this one I really like, is that other people in the context of pancreas have proposed that you generate a plexus with lots of loops and interconnections, and then it's fluid flow that rationalizes the final network that you generate, a bit like what happens in vasculature. And you know, in the case of mouse pancreas, we think it's branching morphogenesis, but we do think that fluid flow plays a key role in remodeling the tissue late on. So, you know, there are lots of other options available. Okay, so in summary, you know, what do I claim that we kind of achieved here? So if we look at the tissue scale of these different branched epithelia, we think that the large-scale organization is reasonably well predicted by this local self-organizing principle based either on this branching annihilating random walk or a variant of it, this inflationary branching random walk. So it's a purely local kind of paradigm in which tips stochastically branch, they elongate, but they annihilate when coming into orbit of a maturing duct, crowding, secreted factors, and so on. At the cellular scale, although I didn't go into details, what we see in pancreas, in the salivary gland, and in the mammary gland, we find that the progenitors in these tips function as equipotent populations that are lineage restricted to the different sublineages in the tissue, and they act cooperatively to drive these serial rounds of ductal elongation and stochastic branching. And what I didn't go into details in is that there's lots of cell movement during that process. So each time these tips bifurcate, there's lots of cell rearrangements in the tip. So why do we think this is useful? We haven't gained any insight, or at least what I've shown you, into molecular mechanisms. But we think that this is important to understand because it gives us a quantitative platform to begin to understand what are the niche factors that control self-renewal at the tip, the lineage restriction of the progenitors at the tip, the mechanisms that balance duplication and differentiation at the tip, and the mechanisms that control stochastic branching, and finally the factors that drive arrest or delay when these tips meet maturing ducts. So that's it. I think I acknowledged pretty much everyone. Actually, Magda did the pancreas work, which I didn't show, but I highlighted, I think, everybody else. I want to mention especially Edouard. So the way we got into branching at all was through Edouard's intervention as a postdoc in the lab. And all of those early ideas, he was just, you know, I can't overstate it. He was instrumental in really driving all of those kinds of concepts and ideas. He's really a star. And then this work wouldn't have been possible, I think, without Edouard beginning all of that. So I'm done. I'm finished. Thank you.
Thank you.
Great, thank you for the fascinating talks. We had a bunch of questions, but if there's 5 more questions from.
One more last question. It should be a quick one. So here we focused on like branch epithelia, but there are a lot of other networks, kind of ubiquitous everywhere. So would you like to comment on, like, say, the topology of neural networks? How would it be different from like branch epithelia?
Yeah, no, that's a really good question, and let me. I'm sorry, I'm not going to. Trust me, I'm not going to give another talk. Just a bit of. No, really, I'm not. But I was going to have a. Yeah. So there's lots of branch stuff, right, and including neurons. So you asked about neurons. What I can say is that there has been historically a lot of work on the branching of neuronal structures, and following our work, people have now started to try to think about the problem in the same kind of way, asking whether there's a kind of mathematical branching paradigm that could be described by statistical principles and so on. And Edouard himself actually has uploaded, may even be published now, a very nice paper on the bioarchive that has examples of that. I know that Joe Howard at Yale has done beautiful work in this area and is picking this up as well. In the case of neurons, it's more subtle than what I showed, because in most neuronal contexts, the development of the pattern occurs through a process where they can grow and retract. Right. So mathematically, we can still find ways to describe those kinds of processes, but you kind of move 3 steps forward and then 2 steps back, and 3 steps forward and 2 steps back. So that's the neuronal context. In a similar kind of thing, I think, would happen in plant roots, where they grow and retract when the environment is not so good to generate patterns. Whoops. But I've not seen good data on plant roots. But the mischievous remark I was going to make is what you can see here are trees. But these trees are actually fake. We wondered about trees. So first of all, tree branching is not dichotomous branching. We didn't realize that and had to be corrected by the plant scientists. But these trees aren't real trees. These trees were generated by Pixar. And when we looked at the way that they did that, actually they come from this group in Canada, which then set up which helped to set up the animation of trees in Pixar. When we looked at the way they generated these branching rules, the principles are very similar to what we ended up having to rediscover. So they anticipated a lot of the things that we got to, years before, right? So, simple principles: competitive advantage of main stem limits growth of lateral branches, resource allocation limited by distance from root and competition for light and space, branching, shedding with age. You put in these simple guiding principles and you end up with things that look a lot like trees. So this is the same model, but where you subtly adjust different parameters. So I think the principle very much applies there, but it's not a new thought. This group this is where this group has been doing this for years and years and years. So yeah, so I think, I think this way of thinking should be very effective, and has already been used in other contexts, like vasculature. People are thinking in these kinds of ways, but in other contexts, I don't think people have started to think in this way. I think it's very profitable.
Maybe this could be a final question as well. If I look at kidneys, for example, can I tell the difference between a left kidney and a right kidney just from looking at the branching pattern, some subtle chirality or something?
Don't know. I don't know. That is a good question. Of course, naively, I would say, you know, the prediction I think is no. The prediction I think is no. But yeah, it's interesting. We've got data from Rosemary Samponia. I'm not sure whether she scored left and right. I'm not sure she scored left and right. I can tell you the fun thing, by the way, David, since you asked about the kidney.
Yes.
One of the things that we really wanted to do was we wanted to find a biological way to cross the phase transition from being a growing kidney to a kidney that terminates because it's on the other side of the phase transition.
Yes.
Right, where you terminate on yourself before you ever get going. And there are mouse mutants which have that phenotype. And we would like very much to segment those kidneys which terminate early. The embryos are not viable. The kidneys terminate early to see whether what's happened is that they're not branching enough at a high enough rate so that you cross the phase transition boundary. That would be a really nice demonstration that, you know, that this is a kind of phenomenon taking place. But yeah, the chirality, it's a great question. I don't know. Can
I ask one last question? Sorry. So I know for vascular network, there are many ways to get that pattern through the optimization of the nutrient delivery efficiency. Did you test your network pattern like with any kind of optimization?
Sorry, was this about vasculature?
Yes, I know that some work from Alchemy categories work. Yeah, that's for optimization, but they produce some network pattern. I don't know if your network pattern or topology has any advantage of this optimization.
Yeah, so let me, I can answer this very quickly. So we did nothing on this. So vasculature, to my knowledge, develops as a plexus. There are lots of loops, and then it's thought that the blood flow is rationalizing all of those loops and converting it into the network. At least that's what I understand. So I gave this talk recently in Oxford, and Philip Maney and colleagues told me a little bit about the work that they'd done on vasculature that has a lot of overlap. I mean, earlier than us, that has a lot of overlap with what we've been doing. So I think that the details, of course, are very different because the methods and the mechanisms in which they develop are different. It's this plexus and then remodeling. But the way of thinking, I think, may still be quite valuable in terms of this kind of mean field way of description. And they seem to be all over that. But I suspect, I can't see who asked the question, but I suspect you know much more about that than I do.
Thank you.
Great, I guess, any from this Zoom, I guess? That's not, so I guess, just thank you, Ben, again, for this very nice.
No, thank you, Ben.
Thank you.
Right, it was wonderful. Thanks again, and thanks for putting up with all the questions.
Yeah, not at all. Thanks a lot.
All right. Take
Bye.
care.